Well, let's get started. So today, well, other people are still tuning in. Today we are fortunate to have Dr. Sharlene Santana to give us a talk as this quarter's a science seminar series. And Dr. Sharlene Santana was a Venezuelan native and got her bachelor's degree, I believe bachelor's degree, right? Licenciada from Universidad de los Andes, if I pronounce correctly. My Spanish was terrible. And then she got a PhD at the University of Massachusetts in Amherst. And then since then, that was 2010. And since then, she got a appointment at the University of Washington in 2012. And from there, she started with an assistant professor all the way to professorship. And currently is a curator of mammals at the Berkeley Museum of Natural History and Culture. It's a new facility. I suggest everybody who have time should go to take a look. It's a wonderful facility. And she has published nearly 60 scientific papers in top journals in genetics, ecology, and evolution, especially in the kind of integrative approach to bats. She's a leading expert in the world. She's also a mentor of many graduate and undergraduate students, the whole list of them, lots of them. So that is also greatly appreciated, especially in schools like ours. That's wonderful. Now I let Dr. Santana to give us her talk. By the way, I just to say one more again. This seminar is recorded. All right. Go ahead, Dr. Santana.
Sounds good. Thank you for that wonderful introduction. Let me see, I'm gonna share my screen. Oh, interesting. Okay, just give me 1 second. I need to update my security thing so it will let Zoom share the screen. Okay, I think it should work now. Sorry about that. New computer. Okay. Let me see. Okay, can you see my screen now?
Yes.
Yes, okay, great. Let me just close this. Perfect. All right, so it's a pleasure to be able to talk with you and tell you a little bit about the research that we have been doing in the past few years on bats, all sorts of different topics. But before I dive in, I wanted to give you just a quick overview of the kind of things that we do in my lab as an just general introduction. So we're very interested in understanding the sources of biodiversity. I do a lot of work in the tropics of Central and South America. And it is very obvious there that biodiversity is thriving, it's important, and how complex it is in terms of form, function, and the interactions that are happening between species. So we're very interested in two major questions related to biodiversity. First, how do form and function evolve and adapt in responses to ecological pressures? And second, how does this evolutionary process facilitate or constrain the diversification of lineages? So how do we get many species and what are the factors that are driving those speciation events? And so the lens that we use to answer these questions is a very integrative type of approach. I like to call it evolutionary ecomorphology. So looking at the relationships between morphology, behavior, and ecology in an evolutionary context. And we integrate various fields like morphometrics, biomechanics, behavioral ecology, and phylogenetics. We do this work on mammals, and we've done work on several mammal groups, but bats are one of our favorites because they are incredibly diverse, not only in terms of number of species with over 1,400 species worldwide, but also incredibly diverse in terms of their ecology, anatomy, and behavior. And so when we do these studies, we also not only do it in a comparative context across different species, but also at different levels of organization. So we like to look at different stages of development throughout ontogeny. We also have done comparisons across populations, across ecological communities, and also across different clades. And all these different levels gives us some understanding of how these different factors, morphology, behavior, and ecology sort of had driven patterns of diversification. And so today, I'm going to focus on this sort of bigger level, at the clade level of our studies, looking at how certain factors might drive ecological and species diversification. And we're also going to take a little close look at how some species interactions might drive the diversity of certain traits. So in the first part, I'm going to focus on the evolution of skull morphology across bats. And in the second part, I'm going to focus on the ecological and evolutionary interactions between fruit bats and pepper plants. So in this first part, looking at the diversification of the bat cranial apparatus, specifically the skulls, this is a project that was funded by NSF, and I want to first acknowledge two amazing former postdocs in the lab, Jessica Arbert and Abby Curtis, who did a lot of the work that I'm going to present today. And in this project, we were interested in understanding the factors that have shaped the evolution of cranial diversity in bats. And hopefully you can appreciate some of that cranial diversity by looking at these 3D models of bat skulls. There's a lot of variation in terms of the shape of the skull, how long the snout is, how the snout is turned upside or downside, and so on and so forth. So to understand why bats are so diverse in terms of their skull morphologies, we like to think of the skull as a structure that has multiple functions. So the skull of bats and our own skulls have to do a lot of different things. Our skull protects their brain. It allows us to feed. This supports other aspects of our sensory system, like our eyes, our noses, and so on and so forth. And bats are really great for understanding how all these different functions have shaped the diversity of skulls, because they have a lot of diversity within each one of those functions. So for example, major bat clades differ in their sensory modes, and also within some sensory modes like echolocation, there are also some differences across those major bat clades. So here is a tree at the superfamily level of bats, which I'm going to use to illustrate some of the major trends in sensory ecology of bats. So it is thought that the ancestor of bats was able to echolocate. That is, that they use the echoes from high frequency sounds to navigate their environment and potentially find prey. But this ability to echolocate was lost in the family Theropodidae, which are the all-world nectar and fruit-eating bats, which instead use vision and olfaction to navigate their environment and find foods like fruits or flowers. And there's one genus within this group that has evolved a different type of echolocation called lingual echolocation. And so they use clicks of their tongue to produce sounds, and then the echoes from those sounds to navigate spaces like caves. And the majority of bats are able to use their sense of echolocation, but that sense of echolocation sort of comes in two flavors. On one hand, we have the nasal echolocating bats, which produce sound using their larynx, but the sound comes out of their faces through their nostrils. And usually they have elaborate structures around their nostrils to direct that sound. And that ability to echolocate nasally has evolved several times in bats in the superfamily Rhinolopoidea and in the family Noctilionoidea. And in this family Noctilioidea, we also have a lot of other sensory modalities in addition to nasal echolocation, like oral echolocation, which is the most common type of echolocation across bats, where the sound comes out of their mouth. And also olfaction, vision, and even infrared thermal sensing in the case of the vampire bats. So a ton of diversity in terms of sensory modes across these animals. Now, at the same time, bats are very diverse in terms of feeding function. So dietary diversity is very high in bats, but it's also unevenly distributed. So the majority of bats are either exclusively animalivorous, meaning that they feed on animal prey, like insects, sometimes vertebrates, or they eat plants. So like the theropodid bats, the old world fruit and nectar eating bats, they drink nectar from flowers or they eat fruit. And then again, we have one particular group of bats, the Phylostomidae, which is within that superfamily Noctilionoidea with a lot of sensory diversity, which has evolved all diets that you can imagine that a bat could have. So you have insectivorous species, carnivorous species, omnivorous, nectarivorous, frugivorous, and even the vampire bats in this family. So there's a huge adaptive variation here. So based on all this, our question becomes, how has sensory and dietary ecology impacted the evolution of skull shape across bats? So in order to answer this question, we have to, of course, take a lot of measurements from bat skulls, and we use museum specimens for that. And we're thankful for all the museums that have supported our work. The analysis that I'm going to present to you today span over 200 bat species, covering all families and all diet and echolocation types. Now, because bat skulls are generally very small, we have to use some technology in order to measure those skulls. And the technology that we use is called micro CT scanning. So computed tomography, this is like a CAT scan based on X-rays that we use to create images of the skulls of bats, and with those create 3D models that then we can use to take our measurements and quantify differences in shape. To quantify those differences in shape, we use a method called geometric morphometrics, which is a method very commonly used to quantify shape of biological structures. And that is based on placing landmarks, which I hope you can see here, which allows us then to analyze the variation in shape independently from variation in size. When we run this analysis, we end up with a lot of variables to describe the shape of structures. And as you can see, we have a lot of specimens and a lot of species. So then we apply multivariate analysis that allows us to summarize those big differences in shape into fewer variables that we can use in evolutionary analysis. So in the results that I'm going to show you, I'm going to focus on those sort of variables that summarize shape. And here's an example of a morphospace from our data set, where the first axis here, the first principal component, is describing variation in one particular aspect of shape primarily, which is the elongation of the rostrum or the snout. And then the second principal component is describing the variation in terms of the angle of the snout with respect to the brain case, from species that have an upturned snout to species that have a downturned snout. So when I refer to these variables, just keep in mind that they're composite of the whole shape variation, but that they are describing one major aspect of skull shape. So then with these variables at hand, we can then go to the evolution sort of part of the question, which is, what are the patterns of this evolutionary process in skull shape across bats? And to do that, we use some math. We run phylogenetic analysis. So these are analyses in which we are taking our data and looking at the patterns of those data and then comparing it to specific models of evolution or patterns of evolution. So we compare our data with what is called a Brownian motion model of evolution, where in this case, the skull shape variables will be evolving sort of in a random sense. And then we have another type of models called Orsten-Uhlenbeck models, in which the variables, in this case, the shape of the skull, will be evolving towards different selective regime peaks, meaning that the morphologies are adapting to different potentially ecologies or sensory biologies. So we're looking at which one of these models fit our data the best. And to do that, we use this automatic detection algorithm called L1OU. So you will see in the next slides this little acronym popping up. And And what that algorithm is doing is detecting along the evolutionary tree of bats where this divergence in selective regimes is happening in skull shape. And hopefully this will become a little clearer when I show you some slides with some results, which is what I'm going to do next. Okay, so here are some results on our evolutionary analysis of skull shape, and it's separate for the cranium and for the mandible here. And so I know there's a lot packed in this slide, so I'm going to walk you through what these results are showing. So we have the evolutionary tree of bats, the phylogeny, and these are trimmed for the data that we have for the cranium and from the mandible, respectively. And then you have these asterisks here at different nodes in the phylogeny, and those are representing those adaptive shifts detected by this L1OU algorithm. Again, these are places where the shape of the skull is diverging among different clades. And the different selective regimes are shown in different colors. So for example, if there is convergence in certain shapes, you will see the same color kind of popping up across clades that are distantly related. All right, hopefully everyone is with me until now. And so I'm going to just describe some major patterns that we see after we run this analysis. So one first thing that we notice when we look at these results is that there are a number of shifts that are occurring very early in the evolutionary history of bats, and that is between 58 to 34 million years ago. And those shifts are sort of more supported for the cranium data set from a statistical standpoint than when we look at the mandible. And then there are a series of shifts that are a little later in the evolutionary history of bats, and that they're only concentrated within this family, the Phylostomidae, and they're shown for the cranium and for the mandible sort of in parallel. They occurred within the last 26 million years. And as you might remember, this is the family that has a lot of dietary diversity. So we're going to get to that in a minute. But first, let's look at these shifts that occur early in the evolutionary history of bats. This was a really surprising result for us because we didn't expect to see all these shifts early. And at first, we weren't quite sure why these shifts might have occurred until we started looking at those transitions in echolocation mode across the different clades of bats. And when we did that, we noticed that, wow, these shifts actually match pretty nicely those shifts in the evolution of echolocation strategies in bats. So we see a shift that is associated with the loss of laryngeal echolocation in the theropods. We see shifts that are associated with the evolution of nasal echolocation in several of the clades. We see a shift related to oral echolocation, and as well as some facultative types of nasal and oral echolocation. So it seems like these are shifts in sensory biology are major drivers of the early evolution of skull shape in bats. And each one of them is sort of related to differences in different aspects of shape in the skull, which is what this diagram here is showing you. Long versus short skulls, the rostrum turned up or down, and the skull being tall or being flat. But I know this is a lot of data to sort of look at in detail for the time that we have. So I'm going to give you the punchline in terms of what are the major trends that we see associated with different sensory modalities. So basically what we see is that for the non-echolocators, the rostrum or the snout is sort of in line with the brain case. So this is, I know this is a weird skull. So this is the front of the skull. This is the back of the skull. For the oral echolocators, we see that the snout tends to be pointed upwards. And then for the nasal echolocators, the snout tends to be pointing downwards. And this is a feature that has evolved multiple times across the evolution of bats. Now, after we saw these trends, we were thinking, okay, how could this help with echolocation function? And we don't know yet, but we have some ideas that maybe, in particular, the orientation of the rostrum might have something to do with how the echolocation call beam is being directed. So oral echolocators have upturned snouts because that increases sort of the width of the echolocation beam. And nasal echolocators might have these downturned snouts such that the nostrils are positioned more ventrally and that echolocation call beam might be more in the same direction of flight of the bat. But this is something that somebody needs to test experimentally. So there's a nice project that somebody could do in the future with some live bats and some good microphone arrays. Now, another cool trend that we saw when we sort of split the bats in terms of their echolocation mode is in terms of the diversity or disparity in the shape of their skull. So we found that the non-echolocators, which are shown here in black, tend to be less diverse in terms of their skull shape than the echolocating bats, the oral emitters, and especially the nasal emitting bats. And I know you might be noticing, well, there are fewer species of non-echolocators, so maybe that's why. But we ran this analysis by taking that into account, and we still found that the non-echolocators tend to have less skull shape diversity compared to the echolocators. So that suggested a really interesting possibility that we tested with our data, which is that maybe there are some internal mechanisms that allow skulls to become more diverse or constrain the diversity of skull shape that also occurred in tandem with these evolution of different sensory modalities. So we tested two mechanisms that morphologists usually think about when we're thinking about drivers of morphological diversity. The first one is allometry, that is the facilitation of changes in shape with size. So shape is changing in tandem with size. And the other one is modularity, which is when a structure sort of is over evolutionary time is subdivided into different parts or modules that can vary and evolve independently. And the result of higher modularity is thought to be a more diverse structure over evolutionary time, again, because having more modules allows all these parts to vary independently. So why not? We tested this with our results, with our data from the geometric morphometric analysis. And we found that indeed, the theropodids, the non-echolocating plant-eating bats, which again are sort of representing the shifts here in the phylogeny, have strong patterns of allometry. That is, a lot of their variation in skull shape is explained by variation in skull size. Whereas when we look at the echolocators, very little of the variation in skull shape is explained by variation in skull size. In other words, theropodids structure their morphological diversity based on size more than they do perhaps on other variables. And the major trend that we see within these non-echolocating bats is that the species with smaller skulls tend to have shorter snouts, and the species with larger skulls tend to have longer snouts, which is a pattern that has been described in other mammals like marsupials and felids, and it's called cranial evolutionary allometry. So this is actually a pretty common pattern across other mammals. It has just been, was not present potentially in the ancestor of all bats, since we know that the non-echolocators are sort of nested in the phylogeny. So that raises the question, why did the theropodids sort of re-evolve this cranial evolutionary allometry? And one potential reason is because having a longer snout allows them to potentially take larger food items. In this case, for the fruit bats, that would be allow them to take bigger fruits. But also having a longer snout potentially creates less constraints in the size of the eyes. So as you can see from these pictures, the theropodids, because they rely a lot on vision, they have large eyes. And having larger eyes, as some previous studies have demonstrated, give them greater visual acuity. And in fact, larger theropodids tend to have larger eyes, not just on other bats, but larger theropodids tend to have proportionate larger eyes. So maybe this type of allometric relationship might be helping them with this sensory function of vision that they have evolved. Now, on the opposite end, we have the echolocating bats. And echolocating bats do not follow that allometric trajectory that I showed you earlier. And that is in part because they have certain constraints that are related to echolocation. So if you are an echolocating bat that feeds on insects, you will need to have or to produce high frequency sounds to detect those small prey, because when echolocation is used for prey detection, the size of the prey that is detectable is highly dependent on the frequency of the sound. So small prey need high frequency sounds to be detected. And then on the other hand, as animals increase in body size, just as there's a physical relationship in which the frequency of the sounds that they produce are lower when they are larger. So if you're an echolocating bat, that really constrains your size evolutionarily. So that might be an explanation why we don't see that allometric trend. What we do see instead is that echolocating bats have a greater number of modules in their skull. Let me just put the numbers here. So when we look at the modules that all bats have, we have 5 modules. The non-echolocators also have 5 modules. But the oral echolocators have 7 modules in their skull. Surprisingly, the nasal meeting have the opposite. They have 3 modules, fewer modules. So this puts like a little obstacle here in our rationale that's saying that more modules create more diversity. So there might be some other mechanisms that might be acting here when modularity is reduced. And hopefully that'll become clear when I show you this next slide here, when we look at the evolutionary rates of variation across parts of the skull and echolocation modes. So this is again using still our landmark geometric morphometrics data set. And in this case, we calculated how fast different parts of the skull are evolving. So here what you see is sort of an outline of the skull showing you the rates of evolution. Faster rates are in red and slower rates are in blue. The front of the skull is here. Here's the tip of the snout. This is the back of the skull. So you're seeing it in a lateral view. And then in the bottom, what you're seeing is the top half of the skull. So what I hope you can see is that the non-echolocating bats, their skulls have evolved at a very slow rate. And again, the evolution of their skull shape is mostly structured by changes in size. Whereas the oral emitting echolocators and nasal emitting echolocators, they have faster rates of evolution, particularly in the front and the back of the skull. Maybe the oral emitting echolocators are doing this thanks to that greater modularity. Whereas the nasal emitting echolocators, surprisingly, even though they have smaller number of modules within the skull, they still have super fast rates of evolution in their snout. So there's something going on in this module of the snout that is enabling its fast evolution. And we think that it must be related to this new function of sound emission during nasal echolocation. So all these changes that I show you are very focused on those early shifts in the evolution of skull shape. So what about these later shifts that we see in the family Phylostomidae? Well, these shifts in the cranium and the mandible, they map really well with shifts in diet. So we have shifts that are associated with the evolution of morphologies within the blood-feeding bats, nectar-eating bats with these really long snouts, carnivorous bats, and then different types of frugivores that tend to have sort of shorter faces. So that means that all these shifts are really consistent with transitions to novel food resources. And this is not the first time that this is shown. In the past, and many studies from my lab and many other labs have focused on this morphological diversification of the phylostomates in terms of their feeding apparatus and how it relates to feeding ecology. And so here's a laundry list of lots of variables that have been correlated with different diets in phylostomid bats. And so these are basically like, I like to say, like the Darwin finches of the mammal world. They have evolved all these morphologies in an adaptive way to fulfill all these different dietary niches. All right, so to summarize this first part of the talk, we see that bat skull shape has evolved under multiple selective regimes that match ecology and behavior. So early in the evolution of bats, we see that echolocation and emission types, so oral echolocation, nasal echolocation, and non-echolocation really shaped the evolutionary trajectories of skull shape. And then later, we see that diet was the primary factor that drove those shifts in skull shape evolution in the Phyllostomidae. We also saw that these early shifts change internal mechanisms like allometry and modularity that drive morphological disparity. So in conclusion, sensory evolution was a really powerful force that changed the potential for cranial morphological diversification in bats. All right, so now in the second part, I'm gonna switch gears. I know you are a diverse crowd, so I wanted to show each one of you a little bit of the different things we do in the lab. And I'm going to talk about this other project that we have been working on over the past several years. And as I hope you can gather from the first part of the talk, the evolution of frugivory was really a critical step in the evolution of some bat lineages, like those theropodid bats and how they changed their sensory ecology. And even within the phylostomids, we see the switches in skull morphology too that are associated with frugivory. But also from an ecological and a conservation perspective, fruit bats are really critical members of tropical ecosystems. They disperse the seeds of hundreds of plant species, many of which are pioneer plants, and therefore they are helping to regenerate forests. So I have personally become really interested in studying a lot of aspects of the ecology, behavior, and evolution of fruit bats and their plant resources. And this project stemmed from that interest. So with collaborators here at UW and also from Sunny Sunnybrook, some years ago, we wrote this NSF grant, Chance and Necessity, Adaptive versus Non-Adaptive Evolution in Plant Frugivore Interactions. And we were focusing on the ecological and evolutionary dynamics between the short-tailed fruit bats, which you can see here. These are bats of the genus Carolia, and their favorite food resource, which are the fruits from pepper plants of the genus Piper. And this is the same genus as black pepper. So they're spicy and aromatic fruits. Now, if any of you have ever been to the tropics and caught bats, you would know that Carolia are very abundant bats. They're major consumers of these Piper fruits, and they are effective dispersers of the Piper seeds. So because of this, they are part of a system of mutualistic interactions. One thing that is really interesting about these bats is that even though they're able to echolocate, they are part of that phylostomid family, they find these fruits primarily through olfaction. But very few studies have really looked at, for example, what chemicals from the fruit scents these bats are using to find fruit, how they affect the dietary ecology of the bats. And also, since they are a mutualistic system, we also could think about the potential for coevolution between these bats and the plants that they feed on. And so in this project, we want to take a look at all of those things. We wanted to understand how the olfactory capabilities of the bats coded in the olfactory receptor genome and their behavioral preferences and their anatomy sort of drive the selection of different fruit resources. And in turn, how these plants have evolved different fruit signals and traits that may help attract the bats. So in this part of the talk, I'm going to focus on two main questions within this really broad framework. The first one is, what traits mediate this interaction between mutualistic Carolia and Piper? So we'll take a look at the side of the bat and the side of the plants. And then we'll look, especially in the side of the plant aspect, whether and how these traits have evolved adaptively and co-evolve with the bats in what would be called a dispersal syndrome system. So to answer these questions, we have been doing fieldwork at this wonderful place, La Selva Biological Station in Costa Rica. This is a highly biodiverse site with a very long history in tropical field biology. And we also have this amazing lab facilities there where we can do our work. So in La Selva, there are 3 species of Carolia from top to bottom, and that is from smallest to largest, Carolia castanea, Carolia sowelli, and perspicillata. And in this same order, they specialize on Piper. So Carolia castanea its diet is made up of mostly of different species of Piper. And then on the other extreme, Carolia perspicillata includes other plant genera, And then Carolia sowelli is kind of like in the middle. We also find over 50 species of Piper in La Selva that vary a lot in terms of their habit and fruit morphology. So we have vines, bushes, herbs, etc. And hopefully you can see these are the little fruits. They're actually infrutescences. So they're multiple little fruits, but it's just easier to say fruits. And you can see they vary quite a bit in their morphology, but most of the Pipers that are eaten by Carolia, they sort of look like this. They tend to be still green when they're ripe. And so they don't stand out as much as, say, fruits that are dispersed by birds. Now, one of the also really cool things about working in La Selva is that, again, there has been a lot of studies there, including on this system. And they have already provided some links between these species. So, for example, they have demonstrated that Piper species can benefit from being dispersed by Carolia via greater rates of germination and lower rates of folibrary. So our task here was to first look at what traits mediate this interaction between mutualistic Carolia and Piper. So again, we're going to start with the bat side, and then we'll go to the plant side. So to get at the traits, we first need to quantify the strengths of this interaction. So we spend a lot of time in the field catching bats, collecting fecal samples from the bats, looking at the seeds of different Piper species in those fecal samples, and then quantifying the proportion of the different Piper species. And with these data, then we can run some mathematical models that can tell us whether or not there are certain statistical preferences by these bats on the different Piper species, which is what I'm showing you here. These are some results from a generalized joint attribute modeling analysis. You don't need to know what these analyses do exactly, but rather if you take a look at the coefficients, which are shown in these boxes here, if they are all above or below 0, they tell us that the Carolia species are statistically showing a proclivity to consume certain Piper species or not. So the ones that we find that meet that that requirement are listed here. So we have a few species that are, let's say, not preferred by the biggest bat, Carolia perspeculata, and some other species that are preferred by Carolia castanea. And as you can see, they're the same. So this is cool because it also shows us that there might be some niche separation between the largest and the smallest species. So now that we have these coefficients of sort of preference for different Pipers, we can then link them to the bat traits. So from the same bats that we collected the fecal samples, we also collected head measurements, form measurements, and bite force measurements, and to see which characteristics of the bats we can link to the fruit preferences. And among those, we found that body mass and the length of the form, so basically size, in addition to the species of the bat, explain the presence of some Piper species in the diet of Carolia. We didn't see a direct effect of bite force, which was disappointing to me because I study a lot of bite force. But that's okay. Bite force is also influenced by body size. So there might be an indirect effect there. But we see that size is a major driver in terms of what Carolia species are consuming, which Piper species. However, as you might remember, I told you that these bats are finding Piper fruits based on olfaction. And we recently published this paper looking at the diversity in olfactory receptor repertoires in these bats. And we do find that there are some interesting differences in terms of the diversity of olfactory receptors among these species, with some of them having a greater variability in the case of Carolia castanea versus the other ones. And also the abundance of these olfactory receptor genes in different families. So these are different subfamilies of olfactory receptor genes here in the y-axis, and then the number of those intact genes. And as you can see, for some families, we do see some differences of the different types of genes that they have. So this indicates that maybe these bats are also differing in their olfactory capabilities in a way that would perhaps allow them to find different Piper species differently. Which takes us to sort of the plant side of things, because what bats can find is going to depend on what chemicals the plants are producing. So let's look at what plant traits mediate this interaction between Carolia and Piper. So to answer this question, we spend a lot of time in the field again, collecting Piper fruits. This takes a lot of work because since we're doing a comparative study and the species are fruiting at different times of year, sometimes when it's raining a lot, we have to spend just a lot of time in the field to get these complete data sets. When we collect the fruits in the field, we bag them and we run them through this headspace absorption method, which basically is extracting the air from those bags that contain the fruits and passing them through a series of filters that capture the molecules that make up the scent. And then we bring them back here to the University of Washington, and then we run them through a gas chromatography mass spectrometry setup to identify those chemicals. So long process, just to show you this plot here, which shows you the summary of our chemical results. This is across 22 Piper species and almost 250 volatile organic compounds, or VOCs, that we identify in the sense of different Pipers. Here in this plot, you can see all the different Piper species. We basically just summarize all that information into one graph. And the arrows that you see here are some of the chemicals that explain some of the major variation in the chemical sense, the chemicals in the scent of these plants. A lot of these chemicals are terpenes, which are very common in fruit scents in general and plant tissues in general. And just to give you an idea, just for one species, what the chemical profile looks like and what that might smell like. Here's a little profile for this species that Carolia bats eat a lot of, called Piper sanctifilicis. And it has some of these major compounds, 2-heptanol, alpha-phellandrine, alpha-cubibine, and alpha-carophylline. And these are chemicals that are found in the scent of things like lemongrass, cloves, black pepper, and cannabis. So now that we know the most salient chemicals for these different Piper species, and we know which plant species the bats prefer, our next step was to test which chemicals are preferred by the bats, to then examine how these chemicals might have evolved in these plants. So that involved doing some behavioral experiments in the field inside a flight cage, where we recorded the bats, both in terms of their behavior and their echolocation calls. And we subjected the bats to a two-choice experiment where we had a sample that was spiked with a chemical and then a control sample, which is what you see here. And these are 3D printouts that are shaped the same way as a just general Piper. So I'm going to show you this quick video here, and you'll see the bat coming to investigate the samples. And there he goes. The nice thing is that these bats, they give us a really strong response when they really prefer a sample. They land on the sample, they try to bite it and eat it. And so with this kind of experiments, we were able to determine some of the compounds that these bats prefer. We found that at least 2 compounds, 2-heptanol and alpha-carophylline, are preferred by these bats. And so with those results, and then plus all of our other chemical results, we can then see if the trends that we see in the diet of the bats are related to this chemical preferences, which is what I'll show you here. So again, these are some Bayesian modeling analysis where when the coefficients do not overlap with 0 means we have a positive or a negative response. And so for example, some of those major axes of variation in that sort of chemical space that I showed you earlier, we find that there are certain associations with the diet of some of the Carolia species, and also with the number and diversity of the chemical compounds that are found in the scent of the Piper. For the specific chemicals, we did find that some chemicals that sort of describe the variation in these axes are related to the diet of the 3 Carolia species. So like beta-phellandrine and 2-nonanone. And then of the chemicals that we tested experimentally with the bats, we found that only 2-heptanol was significantly related to the diet of the bats and not the other ones that we found. And this is in part because, for example, this alpha carophyllene that the bats prefer is really found in all the Piper species, except for maybe one. So finally, just in the last couple of slides, I'm going to show you some results of how these chemicals have evolved in Piper and whether or not this is consistent with some adaptation to attack the bats. So we, again, did some evolutionary model fitting, very similar to what I showed you earlier for the skull shape in the bats, testing these Brownian motion models and the Orsten-Uhlenbeck models. And we found that some of these chemical multivariate axes and the total emission of VOCs basically are evolving at random. However, the diversity and the number of VOCs, which are associated with the diet of the bats, do seem to be following an adaptive process. But things get even more interesting when we start looking at the specific chemicals, the chemicals that are significantly related with the diet of the bats, or that we know that the bats prefer in the experiments, which is what I'm showing you here. And again, here, these little circles are the ones that are the symbols that I'm using to represent those shifts in selective regime in these different chemicals. And what we find is that there are shifts that are associated with in the chemical composition of the scents of species of Piper that are highly consumed by the bats, like this Piper peracuminatum, this Piper sanctifilicis, which have also evolved some of these VOCs convergently as well. On the other extreme, we see some species like this Piper evasum, which does not exhibit any shifts and is not consumed almost at all by the bats. And it was really interesting to see this because this is a Piper species that is giant, so potentially not being dispersed by bats anymore. So we see lots of really interesting trends that are suggested that these chemicals might have evolved in tandem with bat consumption. So to summarize this last part here, we found that body size in the bats and the scent chemistry of the fruits are some variables that explain Piper-Carolia interactions. Of course, there are many other variables that could explain why these bats are eating different Piper species. We were able to determine that Carolia prefers certain volatile organic compounds in the scent of Piper, and some of these have evolved as a unique feature of two species that are highly consumed by these bats. And the evolution of fruit scent chemical diversity and the presence of some specific VOCs, like that 2-heptanol, is consistent with a bat dispersal syndrome in some neotropical Piper. And right now we're in the process of measuring other characteristics of the fruits, looking at the habitat of the plants to more fully understand these ecological and evolutionary interactions. And so with that, I want to thank you all for your attention and for inviting me to give this seminar. We have many funding sources to thank, people who helped us in the field, and of course, all the people in my lab. And with that, I will take any questions. I think we have a little time for questions.
Thank you so much. That's a great one, yeah. There's a question. Do you think the Piper species? Oh, yeah, okay.
There you go.
Do you think the Piper species that do not seem to be adapting to the bad chemical preferences are probably reliant on those species for spreading their seeds?
Yeah, that's a great question. I think, yeah, that, for example, in that Piper evasum that has a super large fruit, I think it's pretty evident that it's either, depending on maybe some other type of frugivores, could be birds. It could also just be some passive dispersal. Some of these plants also sort of reproduce vegetatively. So maybe they're using other strategies. And so we've been thinking about this. And last time I was in the field working on this project, we deployed a bunch of camera traps to record at the plants and then see what other animals might be visiting the Piper and interacting with them. And we had some interesting surprises. We've seen some birds consuming Piper, some other small mammals, little mice, some marsupial mouse. So yeah, it's going to be really interesting to integrate those data and try to understand how other animals might play a role in the evolution of those chemicals.
I had a couple of questions. I seem to, a lot of people go down to South America or the South American region for research on animals. Is it just because there's more diversity down there or is it just because it's like different than North America?
Yeah, it could be both. For my work in particular, working on bats, bat diversity there is much higher than it is, say, that here in Washington. And then we had just bats that are fulfilling different ecological roles. So here in Washington, we have some really interesting species of insectivorous bats, but we don't have fruit bats or nectar bats or vampire bats, right? So one of the reasons why I work in the tropics is because that gives me the opportunity to make all those comparisons among all those other species.
Okay, that makes sense. And then you were talking earlier about how there's a bat that has echolocation but uses a different concept to find fruits. Why would it use that different concept over echolocation? Unless I missed it when you were talking about it.
No, yeah, I didn't go into detail. Yeah, so the theropods, which are the quote unquote old world fruit and nectar eating bats, they over evolutionary time, they lost the ability to echolocate. So the general ancestor of bats echolocated, they lost the ability to echolocate. And I mean, why they made that switch is something that, you know, we still will argue about. One thing is that potentially a larger size was advantageous for something else, maybe dispersal or some other aspect of their biology. But like I explained, once you become larger, then echolocation, reliance on echolocation is not great because the frequencies that you can produce are lower. And so at some point there was a switch where they became too large to echolocate. But then because they were being larger, the eyes were proportionally larger, so that could potentially help with that switch to vision instead of echolocation. So maybe there was some selection on body size in those bats, and then that prompted the switch from being an echolocating animal to a non-echolocating animal. I think maybe you're asking also about the tongue clicks too. Is that? Mm-hmm. Yeah. And then so we have that one genus Rosettus, where they live in caves, and then so they just have sort of re-evolved, but a whole different type of echolocation where instead of using laryngeal sounds, high frequency sounds, they use tongue clicks because they just can't use the high frequency sounds because they're too big.
Okay, that makes sense. Thank you.
Yeah, you're welcome. Mm-hmm.
There's another question. Mm-hmm. Just out of curiosity, how quickly are new species of bats being discovered or differentiated in recent years?
Yeah, we still have new species of bats being described every year, especially in tropical regions. There's a lot of cryptic diversity that is out there. Bat species that look very similar to each other, and but when you look at their genes, it turns out they're different species. And then there are also species that maybe behaviorally are also different. So yeah, we're still finding out every year that number. That's why I say over 1,400, because every year it changes. So I just gave up and I just say whatever. And 2 years ago it was like over 1,200.
There's another question here. Probably was related to previous one.
Mm-hmm.
For the theropodidae, mm-hmm. Could it be that they lost the echolocation due to the environment?
Yeah, I mean, there are many aspects of their environment, like what food resources where they're available, where they were roosting, that could have contributed, to the loss of echolocation. It's tricky to answer some of these big questions with evolutionary transitions in bats because the fossil record it's not very informative of like, the loss of echolocation in theropods or even the evolution of flight in bats. So yeah, we have a lot of good guesses.
Or is it possible when they switch the food resources to fruits, and then they don't use the echolocation to maneuver, like they catch the insects?
Right.
Probably.
Right. Yeah, and we see that in the neotropical fruit bats, because they are using olfaction, but they use olfaction to find fruit sort of a longer distance, and then once they get close, they're still using echolocation at close distance. But these bats are also living in very cluttered environments, so maybe that's what maintains echolocation there. So maybe the theropods lived in some more open environments where echolocation wasn't as necessary. Yeah.
I think that answered one of the other questions, that popped up there. So I have a question about the first part of your talk, when you're looking at the skull shape and size, and you kind of superimpose all that on the phylogeny. And I know for a lot of organisms, we're still figuring out those phylogenies. So how much is that shifting, and does that affect the conclusions you're making about the evolution of skull shape in these bats?
Yeah, that's a great point. I think for some of those deeper nodes, I think those have pretty strong support. So I wouldn't see some of those like big patterns that are related to evolution of echolocation to change. I mean, yeah, people are still building phylogenies for bats with like, genome data, whole genomes. There's like the sweep papers can come in, but those relationships tend to be somewhat maintained. It's at the sort of like later branches where things might change a little bit. And then, I could see how, some things within the phylostomids, for example, things could definitely change in where those shifts are, because those are based on what the phylogeny is.
There's another question here. That looks a good one. I noticed that the heat maps of the skulls showed a rapid rate of evolution in the back of skull for some of the bats you studied. Would that be linked to significant changes, improvements in visual abilities? If so, do these bats really rely more on the sense of vision and less on the echolocation?
That's a great question, and good eye there looking at those heat maps. So that rapid evolution in the back of the skull is actually really far back, much farther back than the eyes, and that is a place where some of the jaw muscles attach. And so the temporalis muscle, which is the muscle that we have here on the sides, ours is tiny, but for bats, it just like covers the whole size of their brain case. So that could be related to some sort of changes in that back of the skull and the attachment of that muscle. So I would think that instead it might be more related to differences in feeding on different kinds of insects, like hard insects versus soft insects. But we haven't really tested that because we don't have enough resolution in the diet to sort of classify our species and test it. But I think it's more related to feeding. To feeding, okay.
So you think it's just a muscle thing? You do not think that's the occipital lobe of the neocortex?
Wait.
Change would be.
Yeah. I mean, it could be. Yeah. That could be too. Yeah. I would have to take a look. I'm thinking more feeding because we do see that tidal crest is getting longer and shorter in some species, but you could be totally right.
And also, I personally have a question as to the two of the compounds which look like universal to all these the fruit eating bats. One is heptanol, right? The other is I forgot. The other longer one, but
the carophylline, that's the one that's more universal. The heptanol one we only have found it in 2 species so far, so that's the unique one. Yeah.
So they are universal to all these Piper species because Piper species are the main food for most of these.
Yeah, that's a good point. I think, it's alpha carophylline, and then there are some other ones in those arrows here, other terpenes that are not found in all species, but also most of them, like germacrine D and some of these other terpenes. But so I think that there must be some compounds that are just like general Piper scent, sort of like this is a Piper. It's just like a, you know, if you think about citrus, like something that makes up the citrus scent, and then you have specific chemicals that are telling you, okay, this is a lemon, this is an orange, this is a tangerine, and things like that. And those will be the more unique ones.
Great, yeah.
Mm-hmm.
So I have a question about the diet, since you showed these different preferences of the different Carolia species for the different Piper species and the different chemical compounds. How much do you or others know about how effective these different bat species are at actually dispersing the seeds of the different Piper species?
Yeah, there have been some studies. It's hard, you know, to do it in natural conditions, as you might imagine, in the forest. I mean, but people have done studies of like seed, you know, capturing seeds and that are dispersed by bats, and or look at the germination rates of Piper near bat roosting areas. I mean, at an individual species level, I can't remember a study like off the top of my head where they did like a very controlled experiment with the bats, but in terms of like communities of plants, where communities of plants that have been dispersed by bats, including all these Piper species, there is a positive effect in the sense that because bats are foraging and consuming fruits of different plant species, their droppings can contain different species of seeds. And so the resulting communities tend to be more diverse. And because of that, say in the face of herbivory by insects, then individually the plants benefit somehow from that. So they're experiencing less herbivory rates. And then, I mean, there's also, say, just passing through the gut of the bats seems to also help with germination rates, just in general. But then what happens in the forest is, and then, you know, it's a jungle out there. There's a lot of competition and predation and lots of other things. So it's complicated to like say like the actual fitness of each one of those plants that were dispersed by bats. But so far, I think the evidence that we have points that it's a positive, you know, that is a positive relationship. The plants are gaining something from being dispersed by the bats.
Well, wonderful. Thank you so much for a wonderful talk. And let's give Dr. Santana a warm applause for her wonderful talk.
Thank you so much. Thank you for all the great questions. These were amazing.
Yeah, thank you. And have a wonderful weekend.
Yeah, you too. You all take care. Now you know where to find me.
Yeah, well, I will do that. Thank you.
Okay.
Thank you all.
Bye.
Bye.